morning for some, and maybe good evening for others. I'm Christopher Rose from Université Montréal in Canada, and it's a great honor to introduce our speaker of the day for the 7th edition of Living Liver Seminars. So Sarah Montanesi is our invited speaker, and she's an associate professor of medicine and honorary consultant physician at Padova University Hospital in Italy, and is also a professor of chronobiology at the University of Surrey in the UK. So Sarah has a long-standing clinical research interest in hepatic encephalopathy, which we know is a cerebral complication of liver disease, and has published extensively in the field, and especially in many reviews, and was one of the leaders in the guidelines for hepatic encephalopathy, which was published recently. She's been the past president of ISHN, the International Society of Hepatic Encephalopathy and Nitrogen Metabolism, and is currently vice president and, of course, president-elect of the European Biological Rhythm Society. She's an associate editor and special section editor, Snapshots for the Journal of Hepatology, and associate editor of Frontiers in Physiology, Chronobiology. She's been part of many editorial boards, including Hepatology and BMJ Open Gastroenterology. So as her talk is alluding to today, she has a research interest in sleep-wake disturbances exhibited by patients with cirrhosis, and actually has made a lot of significant contributions in the field, in their understanding and in the treatment. She's currently using her experience in the liver field as a model and applying it to several other areas of medicine. And she's working on the management of hospitalization-related sleep-wake and circadian abnormalities on a definition of patient-specific tools for clinical circadian assessment. She's very well funded from the European Association for the Study of Liver Disease, and as well as locally in Italy, and also the European H2O2. So it's my pleasure to again introduce Sarah Montanesi, and the title of our talk will be biological circadian rhythms in liver disease. So I give the stage over to you, Sarah, please.
So thank you so much. Thank you, Javier and Jordi, for the much appreciated invitation. And thank you, Chris, for the very generous introduction. As Chris mentioned, I'm going to try and discuss with you circadian rhythms and liver disease, a bit about pathophysiology, the diagnosis, and the relevance of what we know about pathophysiology and diagnosis to management of liver disease. All these organisms have a circadian clock. Cyanobacteria, Drosophila, Arabidopsis, Neurospora, and of course, most mammals. And here you see represented Drosophila, Arabidopsis, and Neurospora are the model organisms which have served the chronobiology community best to understand the functioning of circadian rhythms. But the reason I put cyanobacteria in is not only because they also have been an amazing model to study circadian rhythm, but also because they live between 2 and 3 hours. So one could argue, why would something that lives 3 hours need a clock that ticks every 24 hours? And of course, this is kind of dipping into the anticipatory and adaptive value that having a circadian clock has. It is not so important how long you live, but whether you spend these 3 hours in the light or in the dark, in the cold or in the heat, you still need to be able to anticipate and predict that. And while circadian clocks are the ones we know best, I just would like to hint that most mammals and several other animals have, and plants, of course, have annual and seasonal clocks. There are lunar clocks to which, for example, menstrual cycles synchronize in humans. There are tidal clocks. And all these clocks have been shown to some extent to have anticipatory value. There's these beautiful studies about these organisms that live on the beach, very close to the sea. And of course, if they can anticipate the tide, they can retreat before the tides brings them away at sea. So this is what these clocks serve us most with. In mammals, and should you be interested, this is a beautiful review. What we call the circadian timing system, and it really is every day a bit more of a complicated system, is made up of a central circadian clock, which is in the hypothalamus in the brain, and is responsive to a whole set of signals, of which the most important is light, and synchronized to the light-dark cycle dependent on the 24-hour rotation of the Earth. Since 1998, we have known that most organs, and actually most tissues and virtually every cell, have clocks. And while there is a hierarchy within this system, and so the central circadian clocks tend to be dominant and orchestrate all the others, every day we're learning about independence of different clocks and their ability to feed back to the central circadian. Why the liver and the liver clock? Well, I probably really don't need to tell this audience that there are not many ways the liver gets sick. The liver can become fatty and inflamed, and you can have acute hepatitis or chronic hepatitis. And you can go on to have diffuse fibrosis, which then becomes cirrhosis and hepatocellular carcinoma. Up to when I studied medicine and hepatology, there was a dogma that you needed cirrhosis to have hepatocellular carcinoma. And as many other dogmas in hepatology, this has fallen. And of course, we know that steatohepatitis is a risk factor for HCC. And during my talk, I'll try to show you where either central or peripheral rhythmicity is relevant to the evolution of liver disease. The most of what we know so far, and actually what my research has concentrated upon over the past maybe 15, 20 years, has been on cirrhosis. And actually, what I'm going to try and talk to you about is sleep-wake and circadian disturbances in cirrhosis. And then more of the same in view of what we've recently learned that is new about the central circadian clock and about the liver clock. And then I will hint at material of which we know much less, which is the circadian timing system and fatty liver, steatohepatitis, and the circadian timing system in relation to liver cancer. So back to cirrhosis. So actually, chronobiologists, even translational and clinical ones, know quite a bit about what happened to your health if the circadian rhythms are disturbed. But we know very, very little about what happened to circadian systems in disease. And the reason why we know more on this in cirrhosis is a very simple reason. Because doctors became interested in this very early on, because patients would sleep very badly, and they sleep much worse than patients with other forms of chronic liver disease that you could call equally as bad in terms of everyday life or disease impact and so on. And in addition to this, they're very difficult to treat because most common hypnotics are metabolized by the liver and so are problematic to use in patients with liver disease. And as Chris knows better than me, patients with liver disease are extremely sensitive to any form of psychoactive medicine. So they have a big issue that we have trouble treating. And if you actually look at big and small quality of life studies, you know, for all they have ascites and they bleed and they are yellow and they have all sorts of complications. There are two things they always complain. And both of these we are very bad at understanding and treating. One is their sleep-wake disturbance, and the second one is cramps. The first and seminal paper about sleep-wake disturbance and liver disease, besides the original one in the 50s from Sheila Sherlock's group, is this paper. The first author and the last author, both Chris and I knew well, Chris better than me, and very sadly, they both died young. Joan Cordova and Andy Bly. And I'll walk you through these complicated slides. This is a large human studies. You see healthy controls are white squares. Then there's some 50 individuals with kidney failure on dialysis. The one who sleep well are white circles, and the one who sleep badly are black circles. And then there's patients with cirrhosis. The ones who sleep well are white triangles, and the ones who sleep badly are black triangles. Here on the y-axis, you see a morning-evening score. The lower the score, the more of a morning person you are. The higher, more of an evening person you are. The higher the score, the more of a morning person you are. And here, this axis is clock time. And if you look at the picture, you can see there's three groups here with much of a matchness. They tend to have intermediate scores, and they go to bed and wake up at roughly the same times. And these are healthy volunteers and dialysis patients, regardless of whether they sleep well or badly. But what you can see at the extremes here is beautiful. Patients with liver disease who sleep well are much more morning, and those who sleep badly are much more evening, and indeed they go to bed later and they wake up later. And you could, of course, ask me whether this is alcohol-related etiology. A small proportion in this group were alcohol-related etiology, and this has been shown to apply also to hepatitis C and most other etiologies, although large epidemiological studies are still missing. So the more evening they are, the worse they sleep. And they sleep much worse than individuals with kidney failure. So this was a very, very important contribution, because up until then, people just thought, you know, they are so sick, they'll sleep as badly as anybody who is equally as sick. No, it is not the case. So the first thought, again, from the same group, from Andy Bly's group, was maybe the circadian system is impaired. One marker of the circadian system is a hormone called melatonin, which is synthesized in the dark, and so it's high at night and virtually undetectable in the day. And it is interesting to see how there are three studies in which plasma melatonin profiles were studied in these patients. The first one were 6 patients. The one you can see here is mine, as some 10 healthy volunteers and 20 patients with cirrhosis. And then there's a Greek study done in the middle. And although they are performed in very, very different conditions, very controlled, some substantially uncontrolled, they produce exactly the same picture. So in black here, you see the healthy volunteers. This is in a controlled lab. They peak around 2 o'clock in the morning. And here are the patients with cirrhosis in red. And yes, you could argue there might be some problems with metabolism because the area under the curve is a bit larger. But the main thing you see is, no question, is the peak is delayed and the whole profile is delayed. So they start off late, they peak late, and they come down late. And this doesn't suggest the impaired melatonin metabolism in the liver, which we went on to show is substantially normal, except for very advanced liver failure and for very high levels of melatonin. It suggests more like the circadian clock being less sensitive than it should to light, which should suppress melatonin much earlier than it does here. So in these same patients, again, many years ago, in a very controlled laboratory conditions, we went on to perform what's called a melatonin suppression test, or a way to test the response of melatonin to light. In healthy individuals, this is a very, very prominent phenomenon. Melatonin levels go up at night in the dark, and if you switch on a light, ideally a control light, like you can see here, this is nothing like a big lamp where you can control both the intensity and the spectrum of the light you administer. And there's a paint in here that just ensures that the retina is uniformly illuminated, a chin rest and a rest for the head, because the patient in the night has to stay there for some half an hour. Here you can see the half an hour. This was white light at approximately 1,000 lux, which in young adults is expected to result in a 20-30% suppression of melatonin levels. Here you can see the suppression, this is where they started from. These are the healthy volunteers, the same I've shown you before, and these are the patients. Healthy volunteers were recruited to match the cirrhotics, so they tended to be in their 60s. So 25% was an average melatonin suppression level compared to baseline, which is a bit less than a healthy young adult, mostly probably because of issues with the lens, the eye becoming a bit older and the lens a bit yellower. But what you can obviously see is that the response in the cirrhotic patients is much milder, and it lasts much less over time. So yes, they don't seem to understand light as well as they should. So in order to prove that the circadian timing system is not really responding to light, you should show that there are abnormalities in the melatonin profiles, impaired response to light, and also abnormalities in at least an additional circadian marker, which can either be core body temperature or, in the case of what we did, can be cortisol. And these are, again, the same people. I apologize for the change of color. It went to a different paper. And so here, the patients with cirrhosis are black and the healthy volunteers are gray. Cortisol profiles are much less regular than melatonin profiles because, of course, cortisol is also a stress besides being a circadian hormone. So if you wake up the patient, if you hurt the patient when bleeding, then you obtain big jumps in cortisol. So here you can see that in healthy volunteers, there's the expected double morning peak in cortisol, which increases in the morning. First peak at 6 o'clock, second peak at 9 o'clock. And here again, a big delay in patients, maybe a first peak at 9 o'clock and possibly a second, even 2 o'clock. So we've got delays in melatonin and cortisol profiles and abnormal responses to light. So definitely a suggestion of an impaired circadian response to light from the central timing system. At that stage, I came back from the UK, where I did my PhD, to Italy. And I still had a lot of the sort of can-do power that the English system had taught me. So I thought, okay, they don't understand light, we'll give them more. So one of the first things we did was quite a brave thing at the time. We did a clinical trial in which we used one single room in a liver ward as a treatment room, and we had a personalized lighting system which was aimed at giving these people more light and stronger light in the morning with a view to anticipate their timing and increase their response to light. And we use a room nearby as a placebo. We did see something, but probably the can-do was a bit too much, in the sense that we had put ourselves in a very difficult condition, because these were extremely sick patients who, besides their normal insomnia, had the hospital-related insomnia and a whole pile of other reasons not to sleep. So we did more of the same at home in less decompensated patients. First, with what used to be the fashion at the time, which were light boxes, and what is more commonly used now, which is light glasses. And with light boxes, people had to sit in front of the lamp, in this case, early in the morning. With light glasses, especially if they have commitments, they have to work, they can wear them for the same amount of time, which is 30, 40 minutes, while they have breakfast, they get dressed, they brush their teeth, they read the newspaper. So they are a handier tool that serves the same purpose. And here we did better, but again, we found sort of unexpected results. Now, in this instance, we measured urinary melatonin and not plasma melatonin, because, of course, at home, you cannot measure plasma melatonin. So you will see histograms rather than points in time. And you will see single cases on the left-hand side prior to light treatment, and on the right-hand side, in yellow marked here, after 2 weeks of light treatment at home. I'll show you one so we can go through it. So urine is collected in 8-hour periods overnight and 4-hour periods in the day. And although, of course, like I say, you see histograms and not nice smooth points like in plasma melatonin, there is no questions that overnight, it's normally done for 3 nights and 2 days in between. There's very high levels of 6-sulfatoxin melatonin, which become much lower in the daytime here. And in normal people, the peak is moved by approximately 1.5, 2 hours. And of course, the counting and the establishment of circadian parameters is a bit more complicated, and it needs a bit more time modeling, but it's still relatively easy to. And here you see the same person prior to and after treatment, then another case, and then another case. We did 18. And I could show you the averages, and I could go on showing one by one, but it's very obvious that we didn't see the phase anticipation that we were expecting. So the light didn't push the night histograms backwards. But the difference from left to right is, in general, and was in most cases, a much better fit of the sinusoidal wave that we fitted into these data. And this these days has become very fashionable, especially in a society when we're exposed to light at all times, and is now called rhythm strengthening. So you get stronger rhythmicity, although we didn't change the time of melatonin rhythmicity, at least in this group. But it was an interesting result, and it also resulted in some improvement, not in sleep timing, which was still very delayed, but in sleep quality, which is reasonable and expected. Now, of course, I certainly do not need to talk to Chris about hepatic encephalopathy, and you all know that hepatic encephalopathy is associated to some degree of somnolence. It can be associated with sleep-wake inversion. I'm not sure encephalopaths are true insomniacs. I'm under the impression that sleepiness is more of a feature of hepatic encephalopathy. But this is, of course, and other aspects of liver disease and circadian rhythmicity and sleep worth exploring. And it can be explored in relatively controlled condition by inducing hyperammonemia. In this case, we did so by administering a mixture of amino acid simulating the composition of approximately half a liter of blood, which was the original Chris Record oral amino acid challenge, mimicking a bleed, and therefore the hyperammonemia that you get when you bleed from varices. And here you can see these were 10 healthy volunteers and 10 patients with cirrhosis, the healthy volunteers in white and the patients in black. We fed them this challenge at 8 o'clock in the morning. And please don't be frightened. This is capillary ammonia in micrograms per deciliter, so much higher numbers that you used to see. And you can see that healthy volunteers start lower than cirrhotics, but of course both go high, because this has nothing to do with liver disease, but it has to do with the absorption of blood. And then healthy volunteers go down fast, while patients with liver disease with a urea cycle defect, of course, they maintain high levels of ammonia. And what can be observed in response to this challenge is an increasing somnolence. Here you can see only the 10 healthy volunteers on a day when they had standard breakfasts without coffee, and here on the day when they had the ammonia load. And this is a self-rated subjective somnolence scale. And you can see what should happen. One gets up in the morning, they're still a bit sleepy, then it goes down, and then it goes up. But look how much higher it is when they have the challenge. And you can see that the highest, the top level, roughly coincides with the highest ammonia level reached after the challenge. And interestingly, the profile in the patient is very similar overall. But the difference is much smaller, most likely in relation to the fact that patients start higher and they are used to hyperammonemia. So if you load them, they feel it less. They probably tolerate it a bit better than healthy volunteers. Here you can see the same study and the same group of patients, and it's the last slightly complicated slide, so I'll walk you through it. After we did the challenge and we measured ammonia and sleepiness throughout the day, in the evening, we performed what is called a NAP protocol. So it's a polysomnography which is somehow compressed in a couple of hours. You have to hope that the patient fall asleep, and in most instances they did. And here you will see EEG features as the ratio of the day when we gave them the ammonia load over the baseline time. So anything around 1 means the situation was the same. Anything over, higher after the load. Anything lower, lower after the load. And in here, the 10 healthy volunteers are in blue and the 10 patients are in red. And you can see that virtually you get exactly the opposite effect. So may I remind you that both patients and healthy volunteers developed somnolence in relation to the load. But if you look at the polysomnographic spectrum, this is slow wave sleep, so deep restorative sleep. Healthy volunteers get a lot of it. This is superficial sleep, so patients get a lot of it, and healthy volunteers get almost none. And these are sleep spindles, which is the early phase of sleep. Not much happening to healthy volunteers. Patients lose them. And this we were very interested in seeing because it's also something that was observed many, many years back in 1949 in one of the few sleep studies of hepatic coma patients. We don't have a pathophysiology for it, but hyperammonemia is associated with loss of space. So what this graph sort of tells you is that both healthy volunteers and patients with cirrhosis develop somnolence in relation to an acute ammonia load, but healthy volunteers are capable of converting this into deep restorative sleep, while patients are not, and if anything, they sleep worse. We were subsequently able to show the same in a model of more chronic dietary hyperammonemia. Here you can see, again, polysomnographic data, wakefulness overnight, non-REM sleep, and REM sleep on a standard night in white in control and hyperammonemic animals. And after a protocol of sleep deprivation, is a very gentle form of sleep deprivation because you stop the animal sleeping by gently brushing them with like a painting brush. And of course, you can see in the controls that after the sleep deprivation, there's much less wakefulness in black here, more non-REM sleep and more REM sleep. And you sort of see the same in hyperammonemic animals, but the effect is much more obvious, and it is especially very short-lived. You see, after less wakefulness here, then the two graphs overlap, the same here for non-REM sleep, and the same here for REM sleep. And in these same animals, with the collaboration of a nice sleep lab in Finland, we were able to measure in the interstitium by in vivo brain microdialysis adenosine in these animals. And we saw quite an interesting phenomenon. In here, again, the control animals are in gray and hyperammonemic animals are in black. And after the beginning of sleep deprivation, here you can see an abnormally high adenosine response. Adenosine is thought to mediate sleep pressure. So both, again, control and hyperammonemic animals become sleepy after sleep deprivation, but the adenosine response in hyperammonemic animals is much more pronounced. So in some ways, we're starting to see abnormalities in all forms of sleep regulation in response to either cirrhosis or hyperammonemia per se. Now, just briefly, I would like to reinterpret some of these data and show you some new data in light of what we've come to know that is new in recent years, both about the SEN and then later about the liver clock. Now, if you open any physiology book, it says that the suprachiasmatic nuclei of the hypothalamus contains 20,000, in mammals at least, clock neurons. Well, that is probably not true. It probably contains some 14,000 clock neurons and some 7,000 clock astrocytes. These are a set of beautiful experiments performed by groups at Cambridge and Imperial in the UK, who were first able to demonstrate that the central circadian clock is made up of a neuronal clock and an astrocytic clock. And as you can see, for example, from calcium level profiles, the profiles of expression of a gene called CRY1, and some electrophysiological data, glutamate levels, these clocks do not beat at the same time. For example, you see calcium having a peak in neuron at 6 circadian time, and a longer peak in astrocytes at 12, 18 circadian time, and again, disparities in CRY1 and so forth. And so it looks like there are two clocks in the central clock, and they beat at a slightly different times. And by and large, they are out of phase by approximately 12 hours. Now, when we saw this, of course, we became incredibly excited because, Chris knows, again, much better than me, that ammonia mostly affects astrocytes. Of course, for a brain not to work, neurons have to be affected as well. But from a histopathological point of view, what we see in animals and people who die in coma is this big swollen Alzheimer type 2 astrocytosis. And if it's true that we have an astrocytic clocks that beats at approximately 12-hour difference compared to the neuronal clock, hepatic encephalopathy damaging the clock could almost single-handedly explain the sleep-wake inversion that is known to be seen in very severe forms of hepatic encephalopathy. And we kind of produced this during COVID, when one wrote mostly reviews because very little work would be done. We wrote a review suggesting this. You can see here a depiction of abnormal astrocytes either compromising or taking over the neuronal clock. And one colleague from the UK told us it was a brave review, and I was very proud till my husband, who is English, told me that when an English person tells you something is brave, they normally mean you're out of your mind, which was probably true at the time, but still was a very interesting hypothesis, which we were subsequently able to prove. With help from colleagues in the United States who have tools and the technical ability to keep mouse hypothalamy alive outside the animals for as long as 6-10 days, and to measure their rhythmicity, we were able to sort of test our hypothesis. Here, what you see is the oscillation of the expression of PER2, which is the protein period 2, the protein corresponding to the clock gene period 2. And here you see the same oscillation represented in a slightly different way in approximately 450 cells, one by one. And admittedly, here we do not know if they are neurons or astrocytes. It is possible to tell them apart, but at the time we didn't perform this experiment. And there's no question that this oscillation is beautiful, and it maintains its rhythmicity for many, many days. It loses a bit of amplitude on day 5, day 6, but it worked beautifully if you simply put vehicle on top of these SENs. But look what happens if you put ammonia. If you actually have the patient or the interest to read the whole paper, you can see we tested many levels of ammonia, because some of you might immediately argue that 5 millimolar is a very high level of ammonia. There's no question that this resulted in profound desynchronization of the overall oscillation. But look what happened, even after kind of watering this with 5 millimolar ammonia, when we added GAIKI, which is an glutamate receptor antagonist. Yes, you do not go back to the same rhythmicity, you lose in amplitude, but you certainly kind of reverse a good chunk of the effect of 5 millimolar ammonia. I will now show you some videos of exactly the same experiment. Please look at A at the moment, which will start running. See this beautiful oscillation of PER3 expression in the hypothalamic. And this is what happens when you put 5 millimolar ammonia. And this is what happens when you add GAIKI. And the bottom panels simply show you a quantitative measure of what you're seeing in the video, which is the phase, which is very homogeneous in B and F, and it's much less homogeneous in D, sort of depicting the same phenomenon in a slightly different way. So this is kind of reinterpreting ammonia data in view of what we know now about the SEN. And we know a hell of a lot more also about the liver clock these days. As hepatologists, I believe that most of you in the audience will be hepatologists. We know that the liver has zonation, so very different parts of the liver have very different tasks in physiology and metabolism. But we now know that this doesn't happen at the same time everywhere. And if you have a very well-equipped lab, you can model at the same time space and time zonation, largely in the 24-hour, which largely explain why apparently competing and possibly damaging activities can occur in different parts of the liver at slightly different times. In addition to this, we also know that the liver is an extremely powerful clock. The liver, out of all clocks, is the one that resists in the absence of hypothalamic input the longest. And it can be uncoupled from central circadian control by modulating food timing. So if you compress the frequency of meals, so if you feed animals much more often than you would normally do, you can get the liver to free to some extent from SEN control. And you can do the same if you feed the animals much at much kind of longer distance between meals than you would normally do. And this applies both if you administer the same amount of calories within the 24-hour day or different amount of calories in the 24-hour day. So it is about timing. So food timing, and this of course applies very heavily to both cirrhotic and non-cirrhotic liver patients, can affect the connection between the liver clock and the hypothalamic. Another potentially powerful and relevant observation that has recently been made in relation to the liver clock is that approximately 1/3 of liver oscillations, so of proteins that are produced with different peaks at different times and other metabolic and physiological activities, are integrated by the central circadian clock. Here they're represented in pink. Some 15% are independent. Some 18% are driven by food timing and to some extent also by food content. But approximately 1/3 depend on a network of peripheral clocks. These include fat, spleen, the gut clock, and the muscle clock. And the one clock that being disrupted completely compromises network-dependent liver oscillation is the muscle clock. And I think this is an incredibly interesting observation for clinicians, because we know that when a patient starts losing muscle, when they become sarcopenic, then a sort of vicious circle starts, whereby it becomes very, very difficult to recuperate muscle, to recuperate weight, to correct malnutrition. and the patient tends to become more and more decompensating, which is why sarcopenia is a marker of bad prognosis, both before and after liver transplantation. So much so that in a clinical capacities, we've all become acquainted with the appropriateness for prognostic purposes of measuring muscle mass along the vertebra. And we think this is very, very important to predict how our patients will do again before and after transplantation. So I believe all these observations about the relationship between liver rhythmicity and muscle rhythmicity are incredibly relevant to some clinical phenomenon for which we have not only no explanation, but very limited management possibilities. And if you want to kind of have a feel for what we think about this, again, past year, we were able to summarize some thoughts on this in a review. I'm getting to the end of this circadian timing system and fatty liver. We know for a fact that when we are desynchronized, so when our circadian clock and the environmental clock tell different times, for example, in shift workers, which are the best known example of this problem, we sleep badly and we perform badly in the waking hours. But over past years, we've also learned that in this situation, and this for a long time has been, and it still is to some extent, a riddle, we tend to become overweight. We have a much higher risk of metabolic syndrome, fatty liver, and steatohepatitis, besides cardiovascular disease and some types. And this is for a reason that has become clearer in recent years. The liver is actually the one organ where the circadian timing system can tell metabolic status and the other way around. So it is within the liver that the circadian timing system is informed largely by a set of proteins called sirtuins about our energy availability or metabolic status, and our metabolism is informed of what time it is. And when this is broken, this so very important crosstalk, misalignment causes liver disease, and liver disease causes misalignment. Finally, what I was alluding to at the beginning, so the broken dogma of hepatocellular carcinoma needing cirrhosis may also have some circadian roots. Now I'm really going kind of slightly brave, but I'll still show you some data on this, which I find fascinating. This was a study performed in 2018. There are now 4 other studies reproducing this observation. Here you can see a number of states in the United States, and they are colored from red to blue, depending on the distance from the center of their time zone. So the redder you are, the more misaligned east you are, the bluer you are, the more misaligned west you are. And misalignment to the west, so having to push your clock forward, is more difficult for our physiology for a number of reasons. And in this pretty extraordinary study, it is shown very clearly out of some 50,000 cases corrected for anything you can ask for: overweight, hepatitis B, obviously sex, ethnicity, socioeconomical status, and so on and so forth, that if you're under 65, your position within the time zone, so your circadian timing clock being more or less aligned to environmental clock, affects your likelihood of developing hepatocellular carcinoma. And as I say, this has now been confirmed in 4 or 5 other studies, not only for hepatocellular carcinoma, but also for other forms of cancer. Finally, transplantation. Now, we know virtually nothing about transplantation and the circadian timing system, but we sort of should be interested. And there are some groups, a fantastic group in Chicago, that are actually working on this. What are the questions? Well, there are at least 3 interesting questions. First of all, a transplanted liver is a denervated liver, and the central clock informs peripheral clocks of the time both by humoral signs, but also by the parasympathetic sympathetic nervous system. So a transplanted person will not have this piece of information. They will only receive humoral information from the SCN. We know nothing about prognosis in relation to liver transplant timing. This could be, of course, because liver transplantation is much longer than transplantation of other organs. But we do know that both for heart transplantation and kidney transplantation, timing of transplantation affects prognosis. And we also do not know, and this is what people in Chicago are studying, what happens if you give a very evening liver to a very morning person, or a very morning liver to a very evening person. And there are reasons to believe that a large difference between circadian liver clock of the donor and the recipient may account for the likelihood of the SCN coupling that liver, and therefore for short-term prognosis. So I would like to finish by quoting something that we wrote many years ago, almost as a joke, and reading it 10 years down the line, it still seems a bit fun and to apply. Over the last few decades, chronobiologists and sleep scientists have taken an interest in the liver, and they have been rewarded with remarkable discoveries. Hepatologists should try to reciprocate, both for the sake of their patients and their research. There is a liver clock, and it may soon be liver time. And with this, I thank you very much for your attention, and I thank, of course, our funders, and both the group in Padova and the group in Surrey. Thank you very much. Yeah, I mean, acute liver failure is a big issue. What I hinted at is subedema or astrocytic swelling. As far as I know, there's very limited data on what actually edema would do on circadian rhythmicity in acute liver failure, because that's such a complicated situation also to study from a kind of a circadian standpoint. What I always thought could be useful in this respect is there are some people, for example, Dominique Dabout's group in Paris, that have been collected sort of what in future we may be able to use as libraries. They've been collected, metabolomic data, of course, in plasma, in urine, but also in cerebrospinal fluid. And I remember some of their papers with these big tables that they themselves said, we can't interpret them now, but we may in future. And every day there's a paper published on a big journal about metabolomics in over the 24 hours. So I believe in order to study this, we'll have to use what's already there, what has already been acquired, in light of what we are learning about 24-hour rhythms of different metabolites, mostly in lipid and carbohydrate metabolism. Thank you, Tom. This is a very interesting question. I know you're interested. For a while, we thought it could be the retina. I'm not so convinced. What should be said, what I should have said, which I haven't said, is that interestingly, the retinal pineal pathway is essentially a glutamatergic pathway. So I have no question in my mind that it is largely glutamate that impinges, or glutamate deregulation, or glutamate GABAergic imbalance, or the effect of ammonia on glutamate that somehow impinges on this. Even though abnormal responses to light have been observed also in non-encephalopathic patients, that of course still have slightly higher than normal ammonia level. So I think it's neurotransmitter imbalance along this pathway, and more or less of a retinal issue, I think. I may be wrong, of course. This is, of course, an important question. There is no question that if you lower ammonia, for example, sleep-wake inversion reverts completely. I think an area where we should do more and where a lot of information is missing is the whole timing of this phenomenon, because of course we don't have ammonia receptors in the brain. So everything that has been done in humans, we've measured ammonia increases and ammonia decreases in the plasma, but we don't know how long the problem lasts in the brain, within the brain, how long the astrocyte is swollen for, how long glutamate levels and where are abnormal for. And I am trying to get funding to study this. So I think this is very important. There is also evidence from old studies like the one by Juan Cordova and also other studies that also people with very mild encephalopathy and with no encephalopathy at all are insomniacs. So I think we always have to remember that there is a cirrhosis-related insomnia. Then there's now even sleep apnea and all that goes with it for people who are overweight and have metabolic cirrhosis. And then there is an oversleepiness and all that goes with it that is probably ammonia related. So in a whole cirrhotic human being, there's probably, in most instances, 2 and sometimes 3 things at play. And they probably should be studied and understood and treated separately to some extent. It's not about the gut and CNS dysfunction, but there is plenty of data on abnormalities in relation to the pandemic. I don't know that I would call them in relation to COVID encephalopathy, because that would be going a little bit too far. But of course, here, what you're mixing is, again, what the virus could be doing, what the infection could be doing, but also what habits could be doing. There are also plenty of data on not what the pandemic did, but the measures to contain the pandemic did to sleep-wake rhythms and to circadian rhythmicity. Because, of course, we were locked in or out at times, or locked in the hospital at other times. But of course, we were somehow free to express more of our circadian inclination in some instances. For example, we published data on university students during the lockdown showing that there were big changes. I'm not aware of direct information of, if you want to call it COVID encephalitis or brain-related COVID damage and circadian rhythmicity or muscle rhythmicity. In one word, no, I would say, in the sense that I believe that the more what we should aim at doing now is understanding the pathophysiology of this problem and their management. The syndromic approach, in the sense, does sleep belong or does it not belong, has not helped in the past, and I don't think it will in the future. Because calling encephalopathy, insomnia, sleepiness, and everything that goes with it has not been helpful in telling who we had to treat how. So I don't think from a classification point of view it will help. Of course, if we understand how this works in relation to higher, lower ammonia levels of whichever other neurotoxin is involved, or inflammation, which is something I haven't talked about but would be worth a completely different and separate lecture, will help us manage, for sure. I think you raise something I'm fascinated by, because what really chronobiology has, when I studied medicine, we were taught that we respond to the environment, we respond to meal. What chronobiology has taught everyone, but especially doctors, is that we prepare for meals. We prepare for changes in the environment. And of course, we don't prepare 10 times a day. we don't prepare for the evening snack, which I think is the reason why, although it makes a lot of sense, and I'm one of the culprits for writing it in the nutritional guidelines, that people with cirrhosis should eat often and small and should eat before they go to bed. The more I think about it and the more chronobiology they teach me, the less convinced I am that this is going to be the right way. And the more convinced I am that this is why it doesn't work, because it doesn't. It is one of those things that make a lot of sense. But if you look at what happens in reality, it doesn't work. And probably one of the reasons why is that we're not built to eat 5 times a day or 6, and we're not built to eat before we go to bed. They sleep badly, but this all depends on the severity. We don't do much clinical pediatric here, but sometimes we see the youngsters with these problems, and we tend to see very severe forms that end up here because they've had repeated episodes. They tend to be restless more than sleepy, which is slightly different from adults, and could relate to the fact that in a similar way to an ammonia load is better tolerated than a cirrhotic than it is by a healthy volunteer. A child who is used to high levels of ammonia probably tolerates ammonia better. And we see a lot of the few kids we see, there is a colleague in Bergamo who sees a lot of this, tend to be irritable and nervous. So they have the milder forms, because the hyperactive forms of encephalopathy are, of course, less serious than the slow ones, but of course it heavily affects their school performance and so on and so forth.
So I'll just take this time again to thank Sarah Montanesi. That was an excellent talk. I learned a lot again, and it was a great and honor to host you today at the Liver Seminars 7th edition. So thank you very much for attending, and I guess we'll see you next time when Liver Seminars is on next month, I guess. Thank you very much.
Thank you, Chris, and thanks everyone. Bye. Ciao.
Bye-bye.